and I work with Dr. Sudha Rajamani as graduate student, and today I'm going to talk to you about why it is important to consider the prebiotic complexity when you are studying the relevant prebiotic reactions. So I was going to start with the concept of RNA world, but David has already done a very good job, so I'll just skip this slide and we'll go to the steps that are actually involved in the RNA world. So you have this, so how do you get to a functional RNA from a bag of just random chemicals? So you have some chemicals and then supposedly you form ribonucleotides and then they get together to form this RNA, some of them which will be functional. So the next step is once you have this functional molecule, you also need to copy this information very accurately so that you retain the function. So you go on copying these reactions and then you undergo natural selection, and in somewhere in here these functional molecules get encapsulated to give you a structure, something similar to a protocell. Okay, so in the scheme, and I know there are other views about how RNA world could not have evolved de novo, like there are pRNA worlds and so on and so forth, but I'm going to stick to RNA world. And where I work is right here, where I study the RNA replication. So I study how accurately the RNA replicates in absence of any enzymes. So a lot of prebiotic chemists, what they do is they pick and choose their favorite reactions and consider only the molecules which they are studying. So whoever is studying is replication, they only consider RNA, and similar is for amino acids and lipids and so on and so forth. But if you remember the graph or the HPLC profile that Becky showed us this morning from Formose reactions, you have like hundreds of peaks there, and the ribose is the only tiny fraction of the Formose reaction that you get. And similar is the case with Fischer-Tropsch synthesis, like you have thousands of products. So you consider any prebiotic reactions, there is no single product. So the situation might be something similar to this. Like if I want to study lipids, there are also other molecules which are there in this mixture, we just ignore them. But probably we shouldn't ignore them because they might have effects on our favorite reactions. So for example, if you have a molecule which is this tiny, it could probably survive in a pool of large giant molecules. I mean, that might not affect it. But if you have a molecule of similar size, it starts with the diffusion of this molecule gets affected because of you have a lot of molecules in there. And there will be another effects and so on and so forth. So what we decided is, okay, let us add some background molecules to this RNA replication reactions and see what's the effect. So because RNA replication is my favorite reaction, I selected RNA template and primer. So we have this template and we have 4 versions of template because we have 4 bases, and then there is this primer. And so what I do is I add 1 monomer at a time and study the rate of the reaction. So the selected monomer is this, which is a modification at the phosphate end of the monomer just to enhance the rate of the reaction so that I can study it on the laboratory time scale. And the co-solutes that we selected are 2. The first one is a lipid molecule. And why lipid molecule? Because it has implications in forming these protocellular membranes or boundary conditions. And the second one is PEG, because a lot of biochemical studies also include PEG. Because it mimics molecular crowding and so on and so forth. So we selected these 2 molecules. Okay. So how do we study these reactions? So we analyze it using gel electrophoresis and then we get the rate of the reaction. So you have 4 templating bases, 4 nucleotides that come in. So you have 16 reactions and I studied them in 4 different conditions, so 64 different reactions. So let me first show you the results from a matched addition reaction. So by matched addition, what I mean is when you try to add A across a U templating base, that's a matched addition reaction. So there are 4 matched additions and there are 12 mismatched additions. So what happens to the matched addition reaction? So nothing happens to these 2 reactions. So on x-axis you have the incoming nucleotide. So when the incoming nucleotide is a pyrimidine, which is C or U, nothing really happens. But when you have an incoming nucleotide which is G and A, which is actually a purine, the rate of the reaction goes down if you have these background molecules in the picture. Okay, so this might be a bit more clear in the next slide, which is this. So on x-axis what I've done is I've plotted the rate difference between a control reaction where you do not have any background molecules, the reaction is happening in buffered condition, just the buffer, and the second where I have the background molecules. So that's on the x-axis, and I have normalized that on the y-axis because rate of all different reactions is different, like mass additions take place at a faster rate. So these 2 points, they stand out. Everything else is clustered near 0, 0, but these 2 points stand out where you have addition of a purine across a pyrimidine. So yes. So I mean, how does it matter, right? Only 2 out of 16 reactions are getting affected. So what's the big deal? But just now I told you that these are the mass addition reactions. So these are 2 of the 4 fastest reactions in those 16 reactions. So what they do is they affect the number of times you are getting a correct addition to your primer. So number of times you are getting a correct replication. So that's depicted here. So if you consider this graph, anyways the frequency of addition across A and U is not that accurate, and it's already known in the literature. What happens if you start adding these co-solutes? This happens. So the frequency of addition of G across U, which is a wobble pair, it considerably increases when you have these co-solutes in the background, you know. So if you convert these frequencies to mutation rates, you obviously have higher mutation rates when you have these background molecules. So, and just remember, we have added just 2 and not a lot of them. So the effect is really drastic. If you have a U templating base, like it goes from 31% to 51% of error that's occurring. So if you have a functional sequence which is like really U rich, you will, and in this scenario, if it's replicating, it will quickly lose its function because it's not replicating correctly. So that's it. So the main point in this talk that I want to get across here is we need to consider the presence of these background molecules on any of the prebiotic reaction that we are considering, because I just added 2 background molecules and it's actually already is reducing the accuracy of the replication, and so it will affect the way the functional sequences are evolving and everything. Okay, and right now is a good time to do it because we also have very good analytical techniques and everything. So what we want to do next from here is we want to understand why this is happening. So there could be 2 reasons why this is happening. There is a reason why there is RNA and lipid interaction, so we are trying to understand that interaction using some microscopy, and probably I'll have the results pretty soon. But I'm going to share with you the data for NMR. So with NMR, what we want to do is we want to understand the nucleotide stacking. So the hypothesis is because we have this all molecular crowding agents, the nucleotide stacking is getting enhanced in these situations because let me remind you that only purine reactions are getting affected. So purines are known to stack better. So if that stacking increases in presence of background molecules, the purines won't be available for addition to the RNA primer. So that's what we are studying using T1 NMR data right now. So these, and I'm just standardizing these with help of Harshad here in my institute. So this is a T1 relaxation data for AMP, which is a purine, and this is T1 relaxation data for UMP, which is a pyrimidine. So if you see, as you go on increasing the concentration, the relaxation time decreases, which means that the molecular size is increasing. So AMP is stacking better than UMP, and this is just standardization, and we want to, extend this to when we add PEG or when we add DLPC vesicles, what happens, and any suggestions from you guys is welcome because, yeah, this is just fresh. So yes, with that, I think these are my acknowledgements. I thank my professor and lab members, and these are our funding sources, and thank you.
All right, do we have questions?
Hi, is there a reason specifically why you used the non-traditional 3' terminated primer, a non-hydroxyl?
Yeah, so the reason that I've used it is because if you try and add a phosphate to a OH, that will not happen at room temperature. So that's why, because amine is a better nucleophile than hydroxyl group. So that's why we have modified the primer so that I actually can study the reactions in my lab scale, like in 24 hours or so. And this is a very, I mean, a lot of non-enzymatic replication studies, they use this modification.
Other questions? I guess I was just wondering what would happen, because, I mean, obviously the question is always, what if you try other sort of co-solutes and things like that? And, you know, there are limits on time and effort and everything. But what do you think if you were including different co-solutes, or what would be sort of the next one you would want to try that you think would have an interesting effect?
Okay, so the next one that we actually wanted to try and that we are trying right now is replacing the lipid with fatty acids, because that's more like prebiotically relevant. And then later, we might want to go to dipeptides or tripeptides, which actually bind to RNA via electrostatic interactions and see whether they have any effect or not.
Other questions? Oh.
So you are noting that the extra solutes do have the higher rate of mutation, but could that loss of fidelity actually be beneficial towards increased rate of mutation, evolution of life?
Yeah, so there are two scenarios here. If you are already if it is already functional sequence, it is not beneficial. So if it is a functional sequence, it wants to get encapsulated, probably maybe, because then it will be away from all these co-solutes, because if the mutation rate is high, the functional sequence will not get replicated correctly, and because of the loss of information, you might lose the function. But again, it might be helpful in sort of another scenario when you want to get to the functional sequence, like you can probably explore a lot of sequence space. So yeah, it's like it might be helpful in one scenario versus the other.
Okay. Thanks.
Any more?
When you were talking about the T1 relaxation times, I think it was in the last slide, do you think it's possible that the purine nucleotides are aggregating at the higher concentrations? And maybe that's why you're seeing like an apparent decrease?
Yeah, so that's what we are trying to study actually, whether they are aggregating or they are forming some sort of secondary structures or whatever higher order structures. So that's what we want to try using T1 relaxation data, and we are seeing that AMP does show decrease in T1 relaxation. Maybe it's getting aggregated or whatever at higher concentrations, but UMP doesn't. So yeah.
Let's thank Niraja again, and we'll move on.